A very good morning, student. This is Jyoti Pandey, and the today's topic is morphology and classification of bacteria. Let's start. In this slide, we also learn about the eukaryotes and prokaryotic cells and their differences. So, the learning objectives are after completion of this PPT presentation, we are able to learn about prokaryotic and eukaryotic cells. We are able to describe the structure of bacterial cell and explain the function of its various components. And this study also beneficial and gives knowledge us about the why cell wall forms the basis for classification of bacteria. It is also useful for explaining the structural modification of cell and their importance. Now comes to the introduction of bacteria with its morphology. So morphology first character is size of bacteria. The bacterial size can be measured with the unit micron or micrometer. So 1 micrometer is also termed as 10 to the power minus 6, and it is equivalent to the 1 upon 1000 millimeter and 1 upon 10,000 centimeter or 1 upon 100,000 meter. It is also measured in nanometer units. Nanometer means 10 to the power minus 9, which is equivalent to the 1 upon 1000 micrometer or 1 upon 100 and 1000 meter. Bacteria of medical importance, as per their diameter and length, can be measured within the range of for diameter 0.2 to 1.5 micrometer and for length 3 to 5 micrometer. This is a scale which is used for the measurement of size of bacteria. Now comes to the introduction about the bacteria. The bacterial cells is a unicellular cell. So as per the cellular structure, the cells are divided into two categories: eukaryotic cells and prokaryotic cells. The prokaryotic cells are unicellular, and the bacteria comes under the category of unicellular cell. Eukaryotic cells include the example of like animal, plant, fungi, and protozoans. These are multicellular cells. Prokaryote are the molecules surrounded by a membrane and a cell wall. They do not have any nucleus. They do not have membrane-bounded organelles like mitochondria, Golgi apparatus, etc. The large surface to volume ratio. They are having a large surface and less of organelle structure. So the material as a nutrient or the fluid easily pass to the one part of the cell to the another part. This is a structure which depicts the bacterial cell and its labeling. So the bacterial cell outside covered with a three membranal structure. Outermost is which is light skin in color is capsule. Next gray color membrane is cell wall and the yellow color membrane is plasma membrane. These are the three membrane which is present outside of bacterial cell. They provide the protection to the bacterial cell and its inner components. The rest of organs include cytoplasm, ribosome, then plasma membrane, and nucleic acids, plasmids. Outside the spikes-like structure, tentacle-like structure, which is known as flagella, and fimbrin. So some other components are also there, mesosomes and inclusions. Ribosomes are also present in the bacterial cell. So now studied about the function of various components of bacterial cell. Outermost layer, it contains a two-component rigid cell wall, cytoplasmic membrane. Cytoplasmic membrane Inside contain a cytoplasm, a fluid material. Cytoplasm contain a cytoplasmic inclusions, ribosomes, mesosomes, and genetic material. Additional structures include capsule, flagella, fimbriae, villi, spores, etc. The cell wall and cytoplasm. The cell wall is outermost layer, and it is enclosed of cytoplasm, and their thickness of this membrane is 10 to 25 nanometer, and it is made up of peptido. Now the cell wall. Cell wall is the outermost membrane of the bacterial cell, and as per the chemical nature of cell wall, bacterial cell can be divided into the two groups. One is gram positive and second one is gram negative. The gram positive organism contains the lipopolysaccharides as their outermost environment, and the gram negative contains a peptidoglycan as the outermost layer. So, by this cell wall differentiation, we are able to differentiate gram positive and gram negative bacteria. Several antibiotics may interfere with the cell wall synthesis. Then the plasma membrane. The thickness and the function of plasma membrane can be studied. In this study, we know that the cytoplasmic membrane having a thickness of layer 5 to 10 nanometer, and it is a semi-permeable membrane which control the inflow and outflow of metabolic lipoproteins and other carbon. So the other cytoplasmic components include ribosomes, and the ribosomes is a component which is useful for the protein synthesis. Mesosomes is a multilaminated structure, and it is useful for the six of respiratory enzymes. It regulates the processing of respiratory enzymes. Intracytoplasmic inclusion is the another organ. It is reservoir for the energy and useful for the phosphate metabolism. Next component is nucleus. The bacterial cell doesn't contain any nucleus, no nuclear membrane, and no genomic characters. They only have a single or circular double-stranded DNA, which is as a genetic material, which is useful for the transfer of genetic characters from one species to another one. The other organelles are: first one is plasmid, second one is capsule, third one is flagella. Plasmid. Plasmid is an extranuclear genetic element consisting of DNA transmitted to a daughter cell conform certain properties. For example, plasmid is used for drug resistance, toxicity, viscous layer secreted around the cell wall. It is made up of polypeptide or polysaccharide. And then capsule. Capsule is a layer which is antigenic in nature, and it is act as the outermost cell of the bacteria. Then the flagella. Flagella is a longitudinal filamentous structure which is used for the process of locomotion. Locomotion means movement from one area to the another area. Here is a difference between the prokaryotes and eukaryotes. It started with the certain character nucleus. Nucleus is bounded with the nuclear membrane, nucleoli, and chromosomes. Prokaryotic cells doesn't contain nuclear membrane and nucleus, but eukaryotic cells contain the nuclear membrane and nucleoli. Chromosomes: one circular chromosome is present in prokaryotes. In eukaryotes, one or more paired and linear chromosomes is present. Next character is cytoplasmic membrane. In this structure and compositions can be described. Prokaryotes, fluid phospholipid bilayer made cells which is having lack of sterols, and eukaryotes are fluid phosphate by their containing cells, and it also contain sterols. The difference between the prokaryotes and eukaryote can be determined with the help of cell wall also. The cell wall is present in eukaryotic species but absent in prokaryotic species but absent in eukaryotic species. Locomotory organelles, it is present in prokaryotes, and in eukaryotes it is also termed as cilium. Now, the differences about the cytoplasmic component: mitochondria, liposomes, Golgi bodies, ribosomes, and endoplasmic. These are the inner organs which are absent in prokaryotes, but these all organs are present in eukaryotes. Now, these are the references which is used for the further study. Thank you.